me vais a dar muchos seguidores vosotros. Bueno, first of all, sharing the screen. I don't want to do the same as the previous day. And I've got to confess that I'm a little bit lost with you, so you will have to tell me what we've got corrected and what we haven't. Okay, I know that we've been through this, through all this presentation, and I think that the only thing we didn't review was the last part, that is why and how cells divide. Y me faltaba Víctor. ¿Y alguien está 2 veces? ¿O qué? Ah, no, es que sí, efectivamente sois 13, cierto es. ¿Hay fecha de examen? Qué buena pregunta, si hay fecha de examen, para que cuente yo ahora según está esto grabado y ya no podamos decirlo. No, no la tenemos. La tenemos que poner pronto, pero no la tenemos.
El examen será solo de la célula y eso, ¿no?
De esto. El examen será de esto.
Vale.
This that is part of Unit 1 and this that is part of Unit 2.
Bueno, pero me refiero a que todo es sobre la célula que llevamos dado y eso, ¿no?
Claro, todo desde aquí solo, desde esto que llevamos dado.
Y no has puesto fecha de examen, ¿no?
No la he puesto. Yo no tengo problema en ponerla, no me gusta nada ponerla así online, pero cierto es que se nos va echando el tiempo encima y no tenemos fecha. Martes, miércoles, viernes, querría decir que, por ejemplo, para el... No hace falta, dice, no hace falta ni siquiera hacer examen, claro. Que por ejemplo, para el viernes que viene podría estar, pero el problema que tengo con vosotros los viernes es lo de la última hora, que es la clase más corta, con lo cual un viernes a última hora no lo vamos a poner. Podríamos dejarlo para el día 2 de febrero si no tenéis nada. ¿Cómo vais el 2 de febrero?
Bueno, bien. Aunque
Bueno,
hay unos exámenes unos días antes, pero no estamos.
Unos días antes y algunos días después también, ¿verdad? Bueno, os lo pongo aquí y lo pongo con una interrogación. Dice que tenéis examen de inglés ese mismo día. Ah, de la lectura de inglés. Eso ya os lo habéis leído antes, ¿no? Digo yo. Bueno, que
pero
me da lo mismo.
Como un examen de reading, pero con...
One moment, calla, que estoy pensando que, claro, tengo que mirar quiénes sois grupo 2, grupo 1 y grupo 2 y ya se me había olvidado esa cosa. Bueno, que sepáis que va a ser para la primera semana de febrero, ¿vale? Os hacéis a la idea de que es la primera semana de febrero. Ya concretamos el día para cada uno de los grupos. Vale, claro, pues eso. Primera semana de febrero, ¿vale? La semana que viene le damos una vuelta a todo esto, pero no mucho, que luego nos cansamos de tanto repetir lo mismo. Bueno, so we went through these differences between prokaryotic and eukaryotic cells. We talk about the comparison, these very 3 important differences, the nucleus, the size, the organelles, apart from the organisms in which we find it. Evolution, how in evolution we think that the first prokaryotic cells turned into eukaryotic cells and then autotrophic eukaryotic cells. What is the nucleus? What are the 4 parts of this nucleus? And what are the mission of each of these parts? Que sí, pero la estoy haciendo. Lo que tenéis que estar es prestando atención mientras hago las reviews. What is the coordination between the nucleus, rough endoplasmic reticulum, and ribosomes? That's all the beginning of the presentation and the function of the centrosomes. We went through all that the day before. This is the definition of cell, comparison, characteristics of prokaryotics, evolution, components of eukaryotic cells, paying attention to the nucleus and all its parts, and only several organelles, the ribosomes, the endoplasmic reticulum, Golgi body, Golgi apparatus, and the last one, the os, or the centrosome. So we finished with that in the review that we did the day before. And I went very quickly to the activities, so we didn't see this last part, that is why and how do cells divide, which is this last section that you've got here. How and why do cells divide? I told you anyways that this was included in Unit 1 in page number 21, in page number 21. So in this page number 21, you've got, first of all, why do cells divide? And then the second part, how do cells divide? So everything from this page, you've got it summarized in these two sections here. First of all, why? Well, if you are a unicellular organism, if this is imagine this was a fungus, I was going to say bacterium, but it's impossible because it has a nucleus. So if this was a fungus, a type of yeast, una levadura, this cell that forms this unicellular fungus is dividing here. At the end, what I've got is one cell and another independent cell. So because the organism is unicellular, how many organisms do I have here? Carlos, how many organisms do I have at the end of the process? Venga, solo es un número en el chat. How many organisms do I have here at the end of the process? At the beginning, I had one. This cell has undergone a division, and now I have how many organisms? 2. I've got 2 unicellular organisms, so I'm going from 1 into 2. I've got 2 identical organisms. What am I doing? Reproduction. So if you're a unicellular organism, such as protozoans, unicellular fungus, bacteria, they use cell division as the way to perform reproduction. So why do cells divide in bacteria? In order to form more bacteria, in order to undergo reproduction. But does it happen the same in you? We all are multicellular organisms with more than one cell. And every second in your life, your cells in the skin are duplicating, are multiplying. We've got cell division here. Then, does it mean that I'm forming new organisms in my body? Am I doing reproduction? No, I'm not. Why do I want to do cell division? Why do I want to form more cells? Either because I want to grow, so I want to increase my size, or because I've got a wound and I need to restore some structures. So why do cells divide in unicellular organisms? In order to reproduce the organism. Why do cells divide in multicellular organisms? In order to increase the size of the organism, to let it grow, or maybe to replace those cells that die off in the multicellular organism. That is, why do cells divide? And we've got it in page number 21 in your book. The second one is how. And this how is going to take a little bit longer. We're going to see it in the following unit. But by the moment, what you need to know is that when the cell is going to divide, the first thing that the cell needs to do is duplicate the DNA. Here you cannot see it, but this cell does not contain the original amount of DNA as it had when it was working in a normal way, but it has doubled. Bueno, vamos allá. Our DNA when the cell is going to divide is in the form of, what's the name of these structures that form when the cell is going to divide? Does anyone have a microphone that could work, or do you prefer to write it down in the chat? What's the name of this structure that is formed when the cell is going to divide?
Chromosome.
Chromosome, okay. How many chromosomes did we have in our cells? Vamos allá. How many chromosomes do we have in our cells? Any idea? Just say a number.
46.
Perfect, 46 chromosomes. So does it mean that when the cell is going to divide, because I duplicate the DNA, does it mean that I will have 92 chromosomes? No, no way. You are going to form 46 chromosomes, but it is true that your DNA has been duplicated. In which sense? Well, instead of having one structure with only one chromatid, you're going to have a structure with two chromatids. Voy a intentar poneros esto en grande, a ver si veis un poquito mejor. So, so in this structure, there is a chromosome that is formed when the cell is going to divide. In this structure, I've got identical information here to the information there. When the cell is going to divide, all the DNA duplicates, and this part forms that part. This part forms that part. The genetic information in this chromatid, which is all this is a chromatid, is exactly the same as the genetic information in these chromatids. Why does it happen? Because when the chromosome, a ver si me sale, voy a intentar hacer una magia con vosotros. When the chromosome fuses with the aster fibers in the mitotic spindle, remember that the aster fibers are going to join the mitotic, the aster fibers are going to join the chromosome, sorry, are going to join the chromosome in the centromere in this region. So I would have here one centriole, another centriole, that is the one that forms the mitotic spindle, and I will have another one here, ¿vale? And that would be the centriole there. So when the cell is going to divide, these two threads here get shorter. So what does it mean that they get shorter? That they are going to separate this part from this other part as they get shorter and shorter. So this will go to one new cell that will be formed here. This will go to the other new cell that will be formed here. Because this information is identical to that information, the two new cells will have exactly the same information. So when we go here to how do cells divide, which is the last slide here, how do cells divide? Before the division happens, we need a duplication of the DNA. It's not that we have a double number of chromosomes, but it's that each chromosome is going to have two chromatids that are identical. After this duplication, the chromosomes will be separated in two, so I will have an equal division of this genetic material. After the division of the genetic material, we've got here the division of the cytoplasm as well. The cytoplasm will divide later, but it says here that it doesn't need to be equal. I told you the day before, I would say that I told you, yeah, no recuerdo yo si sí o si no, but I would say so. I told you the day before that when you divide two cells, imagine you've got an original cell. This one has, for example, 46 chromosomes like human cells. This one is going to form two identical cells. It's going to form one again with 46 and another one again with 46 chromosomes. But does it mean that I'm going to have exactly the same number of organelles? Not necessary. So imagine that in this cell we had 5 mitochondria there. Bueno, no me van a caber 5, tan grande que lo he hecho. But if they were 5 mitochondria here, like that, ¿he visto qué magias hago? 1, 2, ay, eso por presumir, 3, 4, and 5 mitochondria. When the nucleus divides, it's going to form 2 nuclei at the beginning in each part of the cell. Then I might have this mitochondria 2 in this cell and then 3 in this one. Is it going to be any problem for my cell? No way. Imagine this is a muscular cell, so it needs a lot of mitochondria. Does the DNA contain this information, that because you are a muscular cell, you need a lot of mitochondria? Yes. So as long as you've got all the genetic information that you do have it here, your nucleus will know that this cell needs to form more mitochondria, and you will soon have the mitochondria that you need. You need to ensure that the genetic information is exactly the one that you need. As long as you've got this genetic information,
teacher,
dime.
How they do it? Like creating new mitochondria?
Just use, what are mitochondria made of? ¿De qué están hechas? If you think of the levels of organization that we talked about last year, atoms joined together to make molecules. Molecules that were very complex were biomolecules that could be lipids, proteins, glucides. And then we said that with those biomolecules, we could form organelles. With different organelles, we formed cells. With cells, tissues. With tissues, organs. ¿Os acordáis de esa escala que vimos, verdad? What do you need to form a mitochondria? You just need the right amount of proteins, glucides, lipids, and DNA, because you also need DNA for mitochondria. If you have it, you can form it. So as long as this body is doing nutrition in the right way, it's going to be able to form it. Having the instructions, having the materials, because you do nutrition, then the cell membrane will say, yes, please, I need some proteins. Yes, please, I need some glucides. They will get in and they will form the new organelles. Okay, so the only thing that you need is the information that says you really need it. This division of the cytoplasm does not need to be exactly the same. You will form every structure that you need as soon as you need it. Okay, bueno, pues Then, in this first PowerPoint presentation, you've got these 5 sections. We've already gone through them. If you've got any doubt, you can have a look at the class that we had the previous days. And today, we've got to start with this second part, that are the chromosomes and cell division. Well, today is going to be more chromosomes. The PowerPoint presentation, remember, that is here. And the activities and the next part of the theory is here in this PDF, in this file. Dice aquí, ¿se puede estudiar para el examen solo del PowerPoint? Ay, os he contado tantas veces eso, mírate la clase y te lo ves, ¿vale? From the PowerPoint presentation, you will get the information for especially the first part, the first 5 sections. And then for the second part that starts in page number 4 is this file, what you've got to study. Okay, but in the PowerPoint presentation, you don't have the information on this second part, and is important. I'm sure that I asked you to do some activities, but I've got to confess that I've got no idea which activities I asked you to do, so I need some help. Who is going to give me this help? We already corrected number 1, 2, and 3, ¿verdad que sí? Ay, qué de gente aquí ayudándome.
Sí, hicimos más.
Hicimos más. And I ask you to do from hasta aquí incluso hicimos what is a chromosome. Esa no la hemos hecho con vosotros.
Esa era la tarea, creo.
What is a chromosome? Esta está sin hacer. Describe how structures 1 and 2. Esa sí la hicimos, yo creo. ¿Qué me decís? Si está sin hacer, decírmelo para...
Sí, creo que está hecha.
Está hecha, corregida, vale. What is a chromosome? What is it made of? What is its function? Well, Andrea, if you had to give me a definition for chromosome, what is a chromosome? What would you say?
Profe, es que no lo tengo hecho.
Bueno, pero me puedes decir algo, ¿a que sí? What is a chromosome? ¿Qué me puedes decir? What is it made of? ¿No me sabes contestar a ninguna de las 3?
I don't know, teacher.
You don't know. Bueno. Have you ever seen a chromosome? Maybe in today's class. ¿Te suena haber visto un cromosoma hoy? In today's class. Oh, creía yo que me iba a salir este. No me ha salido. Any idea? No, ¿verdad? Igual no os he contado, no os he escrito, no os he puesto ningún dibujo de un cromosoma hoy. Vamos a ver, ¿qué está? Aquí. This is a chromosome. And we've seen a picture of a chromosome in these slides here. Este era. That is a chromosome. Now that you've got this picture of this chromosome, Andrea, what is it made of? Any idea on what is it made of? Anda valiente. Which type of biomolecule forms the chromosomes? No idea. Bueno.
Oh, teacher, I don't know.
You don't know, pobrecita mía.
DNA.
Chromosomes are mainly made of DNA, but apart from DNA, in order to help the condensation of this DNA, it also has proteins. So DNA and proteins are the components of chromosomes. Second one, apart from what is it made of, that would be DNA and proteins, what is their mission? When are they formed? Chromosomes appear in the cell when? Venga, Lucía, que estás animada.
When they should do cellular division.
When the cell is going to do cellular division, they are present, sorry, aquí, they are present, they appear when the cell divides. As long as the cell is working, in my nucleus, I will have the DNA in the form of chromatin. Chromatin in the nucleus is present when the cell is not dividing, when it's working. But when the cell is going to divide, this chromatin condenses and forms these chromosomes. In the case of humans, it's going to form 46 chromosomes because we have 46 molecules of DNA forming our genetic material. So knowing that we've got all these questions, what is a chromosome, what is it made of, and what is their function, a chromosome could be defined as a structure that is formed in the nucleus when chromatin condenses. So what is it made of if it's chromatin that is condensed? The same chemical composition as chromatin, so DNA and proteins. What is the function of these chromosomes? Well, the formation of the chromosome is going to make this cellular division easier. It's going to ensure that the same amount of DNA goes to each daughter cell. Well, if you remember what I've done with this chromosome, dividing it in two, it was very easy because the fibers that were attached to the centrosome, they only had to pull from this central part, and all this DNA will go to one part of the cell. Imagine that instead of doing this with this structure that is all organized, imagine that you had this chromatin. Chromatin is something like that. This is the chromatin in the nucleus. Well, I'm going to duplicate the amount of DNA, which is the process that happens in the nucleus before the division. Duplicating this DNA means in each thread, I'm going to have another copy of this DNA. ¿Lo veis lo que estoy haciendo? I'm copying exactly the same as I had, ¿vale? At the end I have this. Okay, now separate it, ensuring that half of it goes to one part and the other half goes to the other part. Is it easy to separate it in two? Is it fast enough? Es rápido? Because when you want to separate it, you're going to have the threads that are going to be joined with others, and it's going to be difficult to separate it in a short period of time. So what is the best thing that I can do? Well, once I have all that my chromatin has been duplicated, I'm going to coil, lo enrolla, I'm going to coil all the red DNA, all these new parts, and I'm going to coil this other black part. So I already have, what's that? A chromosome with two chromatids. This one containing exactly the same information as that one. So I don't have a mess anymore, but I've got all the material organized and it's easy to separate it into new cells. Why don't we have the DNA always organized in this way? Because if I need to read the instructions of DNA, if I want to do the copy in the form of messenger RNA, it's going to be easier to read. Perdón, que este me ha salido fatal. It's going to be easier to read this section that is not all coiled. If it's all coiled, it's difficult to read. So when I want to read it and form messenger RNA, because my cell is working, is doing normal activity, I will have the DNA in the form of chromatin. Chromatin is formed when the cell, vaya que 72, no 7. Chromatin is the DNA when the cell is working. All the DNA is not coiled, so it's easy to read. But when I want to do the division, I will form chromosomes. Okay, so that's the difference between chromosomes, which is this structure here, everything organized, this one, or chromatin, that is everything uncoiled, desespiralizado, not condensed. Does anyone have any doubt on this difference? Because it's a very important one. Chromatin when the cell is working, chromosome when the cell is going to divide in order to have all organized and make it easy to separate it in two quickly and ensuring that exactly half of it is on one side, the other half is on the other side, so I can do the division. Okay, that's very, very important. Well, that was the following activity. Is a structure that is formed in the nucleus when chromatin condenses. It helps to make cellular division easier and ensuring the same amount of DNA goes to each daughter cell. Okay, well, that would be the answer for activity number 9 as well. That is asking you for the difference between chromatin and chromosome. Again, chromatin is the form of DNA that is not condensed and is present in the nucleus when the cell is working. When the cell is going to divide, this chromatin transforms into chromosomes. Okay, so you can also do activity number 9, section A. What about section B? Section B introduces another term. Chromatin, again, this DNA that is not condensed, but then this one, chromatid. Very similar terms, chromatin, chromatid. What is a chromatid? A chromatid is, does anyone know it? Probablemente no, because that's a more difficult question. Does anyone know what a chromatid is? Tengo valientes por aquí. No? Bueno, a chromatid is each of these structures that form half of the chromosomes. So it's each of these two identical structures that form one chromosome. All this is a chromatid that is joined by the centromere to the other chromatid. ¿Veis que hay otra palabra parecida? We've got chromosome, chromatin, chromatid, and then we've got centromere that unfortunately is very similar to centrosome and centriole. We've got 3 terms, very similar terms. I'm sorry, but they are the scientific terms for all these structures. So going into your photocopy, you've got all this information. A ver si lo puedo hacer más chiquitito. All this information in page number 4 in your book. We are talking about eukaryotic chromosomes, por cierto. This shape, this common shape like an X, is the shape of a eukaryotic chromosome during one phase of the division, that is the metaphase, one eukaryotic. Then we've got another structure for the bacterial chromosome. Do bacteria have chromosomes? Yes, they do. This is the picture of the chromosome in bacteria. Name of the region in which the chromosome of bacteria is located, the nucleoid is the region of the prokaryotic cell in which I find the only chromosome that bacteria have. Eukaryotic cells have in the nucleus different numbers of chromosomes. It's 46 for humans, but is, for example, 8 for the fruit fly, is only 40 for mice, is a different number for different organisms. But in bacteria, in these prokaryotic cells, there's only one chromosome. And this chromosome is special because it's going to have no end, no beginning. So it's a circular structure in which there's no beginning, no end. Okay? We do have a beginning and an end in our threads of chromatin, and obviously when they coil, they will have a beginning and an end in the structure of the chromosome. But in a prokaryotic cell, it's all that circular structure that is connected. It's all coiled, but it's connected. So that is one important difference between the bacterial chromosome and the eukaryotic chromosome. A ver, que ni tanto ni tan calvo, ¿verdad? Así. Un poquito. So you've got here page number 4 in the photocopy. In prokaryotic organisms, all the genetic material is packed into one single DNA molecule, just one chromosome. The DNA is in the form of a double helix, like our DNA, but forms a closed ring with no free ends. It is permanently attached to the cell membrane. Okay, that is going to help you in the activity that you have to do for the following day, which is the difference between bacterial and eukaryotic chromosome. Pero ahora vuelvo a ello, a lo de la tarea. I will go on with this because I want to go a little bit farther with you, que ya sabéis que we are a little bit late. Eukaryotic chromosome, again, we've got here that the DNA in eukaryotic cells, as we know, is located where? In the nucleus. When the cell is not divided, this DNA is dispersed in the nucleus forming chromatin. We've already seen it. But when cell division starts, this chromatin condenses and it's going to form a specific number of chromosomes, depending on your species. The eukaryotic cell will form this different number. Each chromosome is one single molecule of DNA. And then what is this structure? Because we've got here, si volvemos a esta, sorry, the contents of this unit, unit 5, teníamos aquí que vamos a hablar de chromosomes, the structure, the number, and the abnormalities. So we already know what a chromosome is the DNA that is condensed when the cell is going to divide. What is this structure? We are going to see the parts that form this chromosome. We've already seen one, that is the chromatids. Remember, these two identical components are known as chromatids. This would be one, this is the other one. We also mentioned that these two chromatids are joined by the centromere. That's another name that we've got to study this year. The centromere is said to be a primary constriction. It's a constriction because it gets stretched here. It's not so wide, but it's constricted here. Well, important, this name, centromere, that is this region. It's very important because remember that the threads in the mitotic spindle will join there in the centromere to the chromosomes. And then we've got what we call the arms. In one chromosome, there's 4 arms. 1, 2, 3, and 4. So one chromatid consists on 2 arms that are joined by this primary constriction that is the centromere. we always call the small arm the P arm, and the bigger one is the Q arm. So be careful with that, because sometimes you can see, no me deja copiarlo. Sometimes we see the chromosomes that are not in, that are not like this, like upside in the right position, but we've got them, for example, like this one, this here. So you can see that it has 4 arms, 1, 2, 3, and 4, but who says whether this is the P or the Q arm? Well, it's always, no matter if you find the chromosome like that, or maybe you find it you find it like that, or imagine that you find it like that. Anyways, in any case, the shorter arm is always the P arm. This one would be a P arm. This one is another P arm, and that is the P arm, because the P arm is always the shorter one, while the longer arm is the Q arm. When we represent them in a picture, in those pictures that are a little bit aquí. When we represent them here, we always represent them with the shorter arm. Aquí es que no se ven bien porque son pequeñitos, but we always represent the shorter arm on the top, the longer arm on the bottom. So always like this. Q arm on the bottom, P arm on the top. But the difference is not whether it's in the upper part or the lower part, but the size of the arm. So we've got P arm, Q arm, centromere, chromatids, and then we've got this one that is the telomere. Telo means the end, ¿vale? Por eso at the end of a play we put down el telón, ¿vale? Because telo comes from the Greek term that means the end. So telomere is the region, the section, mere es como una pieza, una fracción. The telomere is the final part. The centromere is the central part. Vale. So that would be another term. The centromere is a very important region in chromosomes, because the older the chromosome is, the smaller the telomere is. The telomere contains information that is apparently unuseful information. Is not going to code for anything. It's not going to have important structures, but it's important because it's protecting the important information. It's like the covers of a book. In the cover, en la portada y en la contraportada of a book, you are not going to find any important information. But if you don't have this cover, the book, the important content could be damaged. That is more or less the mission of the telomere. They're going to be in both extremes of the chromosome, and they are protecting the important information that relays in the chromosome. So it's important that we have them. And we've got here 3 shapes depending on where the centromere is located. If both arms are the same length, metacentric. One of them is smaller than the other one, submetacentric. Apparently we've got no P arm, just a tiny structure there called a satellite, then is acrocentric. And if there's definitely no P arm, is telocentric. Okay, bueno. I've introduced loads of terms today, so we're going to finish the class right now, and you've got some spare time. But before doing that, as usual, all the activities that you've got to do for homework. Your activities are going to be, as I told you, activity number 8, that is just describing the prokaryotic cells' chromosomes and explain the difference between prokaryotic and eukaryotic, is just copying the paragraph that we've read in page number 4 in this photocopy, so not difficult at all. And then you're going to do these pictures from activities number 10 and 11. One picture representing the chromosome with all the parts, chromatids, centromere, P-arm, Q-arm, telomere, every part, and differences between all these 4 types of chromosomes depending to their shape. So you're going to draw a metacentric, submetacentric, acrocentric, and telocentric chromosome. Not difficult at all, these 3 activities, but please do them because I want you to have them in your notebook. Any doubt?